My name is Yonela Tukwayo. I'm a senior researcher in the Energy Center. I am an economics by background. What that means for the speakers is that when it comes to the title of your presentations, you're gonna have to say those yourselves. So presenters, you have 15 minutes for your session to do your presentation. And then in the last 5 minutes, we're gonna do questions and answers. When we have 2 minutes left to your presentation, I'll start making my way slowly to the stage. That simply means that you must start wrapping up your session. In the last 5 minutes, we're gonna have a Q&A. For that, I ask colleagues that we please keep our questions brief, as well as our responses brief, so that we can get home as soon as possible. So with that said, I would like to introduce our first presenter, Ebeni Langa. ICT, please help us.
Good afternoon, everyone. My name is Ebeni Langa. I am a researcher in the Bioprocess Development Group. Today, I will be sharing the project that I worked on in the past 2 years, titled Fungal Mycelium as a Sustainable Protein Source from Development to Pilot Scale Production. As we all know, protein form an important part of the human diet as it supports general body functions in terms of degrading the food that we eat and also provide storage molecules and also assist in synthesizing the hormones that our body needs, as well as supporting our skeletal movements and general immune responses of our body. And having a balanced protein diet generally assists in maintaining a healthy body weight. Here on the right-hand side, I had an example of the foods that we take in our daily diets that provides us with a balanced protein diet, which constitutes animal and plant-based protein sources. However, conventional protein production systems are associated with various challenges that have been reported globally, which means that the current systems do not have the full capacity to provide for the demand that is required with the rise in population that is estimated to be 10 billion people by the year 2050, and the general global shift in health awareness, and the fact that overconsumption of meat is associated with causing non-communicable diseases, which ended up in shifting consumer preferences. And also, with plant-based production systems, there's reported agricultural instability that is caused by climate change issues. And lastly, the livestock production is also associated with negative environmental impacts, such as emission of greenhouse gases. Fungi has been used as a sustainable protein source for over 1,000 years. This includes edible mushrooms that are well known that we have in the market, including button mushroom, shiitake, oyster, as well as enoki. And as of 2002, there was a vegan protein alternative that was introduced that was referred to as, or it is provided by corn, and it is produced through a solely liquid mycelium production process. And in general, fungi provides high nutritional food products with low calories and low fat that are high in fiber, rich in antioxidant, and a protein content ranging between 19 to 55%. In South Africa, the mushroom production industry is dominated by Agaricus bisporus, which is button mushroom, and other species, Pleurotus austratus, as well as Lentionella idotes, are also at the development stages. However, there are various challenges that are being experienced by this industry as these food products take longer to grow. There are high operational costs involved, and the biotechnology strategies, which is mostly through cultivation in liquid mycelium, there is a lack of the infrastructure there in the industry, as well as the skill gaps and the lack of post-harvest infrastructure as these products are highly perishable. Therefore, the CSIR saw opportunities in assisting or developing these technologies, which then constitutes one of our values in localizing commercially relevant technologies, thus contributing to the sustainability development goals in terms of addressing food security issues, being able to implement circular bioeconomy business models, and thus influencing the socio-economic status of our country. Generally, mushrooms are saprophytic fungi as they can grow on dead trees in the forest, and for commercial purposes, they are produced through a process called solid substrate fermentation through use of either grain spawn or liquid spawn, which then feeds to the mushroom solid substrate fermentation. The liquid spawn is known to provide faster growth rates compared to the grain spawn, so it is one of the main biotechnology strategies in addressing the issues that I addressed earlier. And then for mycoprotein, it is mainly produced through liquid submerged fermentation. So then the aim of the study was to conduct development and demonstration of cultivation processes of fungal mycelium for use as sustainable sources of protein. These were the objectives were to select nutritionally and commercially relevant fungal strains with the potential to be used as sustainable protein sources, investigate their cultivability, and to develop scalable production processes up to 30-liter bioreactor scale, and to demonstrate the production technology of the best performing strain at pilot scale to validate the commercial viability of the developed technology, and to lastly assess the performance of the produced biomass in terms of yield, protein content, as well as nutritional profile. This is just a summary of the process of the R&D approach that was taken because to make sure that we get to repeat the results that we obtained at the end of each process, we needed to make sure that each of the starter culture that we have throughout all the research batches that we needed to perform, we have a reliable culture stock. Therefore, we needed to isolate a pure culture of each isolate. We needed to establish reliable and reproducible cell banks. And then we needed to assess the growth of each strain at flask scale to make sure that by the time we move to 30-liter bioreactor cultivations, we have at least established the medium that we're going to use and the growth conditions, which was then optimized as the scale to ensure that we achieve reduced process times. And the developed technology was then moved on to 1,000-liter scale production, which is pilot scale, and we developed different solid liquid separation processes. As in the bioreactor, we would obtain a culture that is mixed with spent nutrients, so we needed to separate what we produce and make sure that the product that we get is a product that we can assess the nutritional content and make sure that it looks like what a mushroom would look like. For establishment of our cell banks, because of the nature of the strains that we were working with, we had to determine to use two different techniques that would be suitable for each strain that we were working with. For instance, for for all 4 strains that we were working with, we tried 2 different techniques, namely agar discs, cell banks, as well as liquid mycelium. For agar discs, we noticed that it was mostly suitable for mushroom strains, as there was a homogenization step that was required before starting the process for these strains. However, this technique was time-consuming and had higher risk of contamination, which is what we do not want for our process, as we needed to have a pure starter culture for each and every process. And mushrooms are very susceptible to contamination. And then luckily for Fusarium venenatum, we managed to see equal reproducibility when both techniques were used. So these are just images of our cultures when we were validating the reproducibility of our cell banks, and they did show to work. Okay, so in terms of demonstrating the cultivability of our strains, firstly, I would like to mention that we observed in the previous step that the Agaricus bisporus strain was still taking longer to grow, which it was about more than 30 days. So for the sake of time, because we only had a year to complete the project, so it was not suitable for that particular time, so we removed it, and we moved on with the 3 strains, which is Lentinella edodes, Pleurotus ostratus, as well as Fusarium venenatum. We selected different medium formulations. For some strains, it ended up having to be about 11 different recipes, but for the sake of this presentation, we selected top 3 that showed to give us acceptable performance. I will start with the Lentinella edodes. For Lentinella edodes, we saw acceptable growth of about above 2 grams per liter of biomass in the medium designated as test 3 in the presentation. And for Pleurotus ostratus, it was about above 18 grams per liter in the media designated as test 2. And for Fusarium venenatum, we got about 29 grams per liter in the medium designated as test 3. All this medium, the differentiating factors were different concentrations of the carbon and nitrogen sources, and they demonstrated variation in their growth rates, with Fusarium venenatum growing within 2 to 3 days, which was suitable for our process and the objectives that we had set. We moved on with the conditions that we had developed at flask scale and 2 30-liter bioreactors. With Lentinella edodes, we still observed similar performance that we saw in flask cultivations. And with Pleurotus ostratus and Fusarium venenatum, we did achieve productivities that were comparable in literature. However, I'd like to point out this biomass yield here. It was mainly due to the fact that the concentration of the sugar that was added in the beginning of the process was a bit lower compared to the Fusarium venenatum. However, the results that we got were comparable to what has been reported in literature in about 3 days. As you can see, most people cultivate these strains for a bit longer, even in bioreactors. Because of resources, obviously, we were not able to go longer than 3 days. And for Fusarium venenatum as well, we observed mostly similar results with 21 grams per liter of biomass that we obtained in 44 hours. For development of our downstream processes, because of the nature of the broth that we would result after the cultivation process, we would recover about 11% of our biomass, which does not mean it's low because of the nature of the mushroom. That's the only maximum yield that we were able to achieve. And then we tested different drying techniques, with freeze drying being best suitable for the Pleurotus ostratus. However, we observed higher losses of the product when this technique was used. And similarly, with the Fusarium venenatum, we got about 11%. And for this one, we tried both freeze drying as well as tray drying. However, we saw that the freeze drying, the product maintained the nature, the similar appearance as the wet biomass, whereas the oven drying changed the appearance of the product, which is also attributed to the different techniques or different processes that these techniques afford drying. The freeze drying uses the drying process, the ice and water molecules are removed from the cells, whereas the oven drying applies heat to the biomass, hence the change in the product appearance. For it was mostly said that the technique that can be selected should be determined by the operational cost, with oven drying being most cost effective and freeze drying a bit expensive. So we also moved on to demonstrating our process at 1,000-liter scale, and this process required a two-stage step, as we needed to start at flask scale, move on to 30 liters to get a seed culture of about 30 liters to transfer to the 1,000-liter cultivation vessel. We did optimize growth conditions here, and the seed was harvested after 23 hours of cultivation. And in the 1,000-liter, we observed a similar performance with the feeding of the bioreactor started after 24 hours. As you can see, we harvested the seed at 23 hours, and when we got to this production vessel, the feeding, which means after the depletion of the carbon source, we needed to add glucose to the vessel. Because also of the nature of the biomass that was being produced, we were not really able to maintain the dissolved oxygen. However, that is in the process of being optimized. And this is the nutritional profile of the product prototype that we generated that were comparable with the commercial benchmark, which is corn, and even the freeze-dried biomass showing a little bit high protein content. In conclusion, we have successfully developed the liquid mycelium production processes and demonstrated the technology at pilot scale, which showcases the potential or capability to localize this technology in South Africa. The growth challenges for other two strains are observed in literature, and further development strategies are required. However, there is a potential application for these in terms of providing the local producers with the liquid spawn, because the grain spawn is still contributing to the longer growth cycles that are observed. And we also recommend the use of models to optimize growth conditions. And the current work is to optimize growth conditions at production scale with potential use of waste streams to apply the circular bioeconomy business models. And the project is one of these will be implemented in the current phase of the project, which is for commercialization under the C-cubed enterprise. I would like to acknowledge my supervisors, and thank you.
Thank you, Tabeni. Well done. Thank you for that very interesting presentation. Colleagues, do we have any questions for Tabeni?
Hi, I'm just curious to know, doesn't the process of either freeze drying and oven drying interfere with the protein capability of the product? Because I would assume like there is denaturing that you're trying to avoid by all means.
Okay. We saw that the freeze drying did not have any negative impacts, but with oven drying, we did see that it had lower protein content compared to the same product that was freeze dried. So the recommendation that I think we have is playing around with the temperature and making sure that at least we dry at about 30 degrees, because it does dry, it's just that it takes a bit longer. Yes.
Thank you, Tabeni. I saw you mentioned your yield, biomass yield, 11%. Does that correlate to your protein recovery? How much protein did you recover?
No, that's not the protein. It's like the physical biomass that we extracted from the broth that after fermentation. It's just the drug, the physical biomass,
So did you measure the
the protein.
protein recovery? No.
Not yet. Oh, good.
Okay, okay. Because I was just going to say that maybe once you've done that, you can compare to other sources of protein to see the performance.
Okay, we'll do
Thanks.
that. Thank
Sharp.
you.
Thank you, Tabeni. Well done. Our next presenter will be Brittany Cedris. If you can just give a round of applause.
Good afternoon. I am Brittany Cedris from the University of the Free State, and my project focuses on valorization of biomass, particularly investigating the enzymatic breakdown of biomass to produce complex oligosaccharides. At the center of the circular economy, lignocellulosic biomass presents itself as a particularly renewable and underutilized product. It composes of three major components: hemicelluloses, celluloses, and lignin. Hemicelluloses are particularly noteworthy for their abundance in xylin. Xylin acts as a precursor for xylo-oligosaccharides. These xylo-oligosaccharides are known to have antioxidant properties which are linked to their side chain substituents. The challenge, however, is the recalcitrance of lignocellulose, which is difficult to break down. This is due to the intricate cross-linking between the polymers. Enzymatic hydrolysis provides an eco-friendly, resource-efficient solution to break down this biomass. My study aims to characterize the C1184 secretome from A. spigillus niger. My study aims to characterize the C1184 secretome from A. spigillus niger, produce and quantify complex xylo-oligosaccharides, and look at their antioxidant properties. For substrate specificity, we examined the C1184 secretome with 1% weight per volume substrates for glucans, xylins, as well as starch, using 50 millimole sodium citrate buffer pH 5. We did the reactions for 1 hour at 37 degrees Celsius. For biochemical characterization of the C1184 secretome, we tested the pHs for a pH range between 4 and 9, temperatures between 30 and 50 degrees Celsius, and the thermostability was tested for hours 1, 5, and 24 hours for temperatures 37, 50, and 70 degrees Celsius. For kinetic parameters, we used the McAllis-Mentum for substrate concentrations ranging from 0.05 to 16 milligrams per mil. A zymogram analysis was done to look at the specific enzymes involved in hydrolyzing this biomass. We did a native page for cellulase, xylin, galactomannan, and starch. the oligosaccharides were produced by doing a 24-hour incubation with the C118 secretome and beetroot xylem. We then did precipitation using organosolvent using a 1:4 ratio solvent solution with ethanol. This was then, the resulting pellet was then dried and resubmerged in sodium citrate pH 5 buffer, where we then did LCMS analysis. We also used this to do further reactions using esterases, so 2 furyl esterases and glucuronol esterase and a GH10 xylanase. This was analyzed using TLC as well as looking at their antioxidant quenching abilities using 2 common methods, ABTS scavenging as well as H2O2 scavenging. For substrate specificity, we saw that the enzyme has multiple activity across xylem, cellulase, and starch substrates, with xylem being higher. This demonstrates that the secretome is a multifunctional enzyme. This was further demonstrated by the zymogram analysis, which shows us multiple bands for the various substrates. We see multiple band profiles on cellulase, xylem, starch. However, for galactomannan, we did not see any activity. For the characterization of the enzyme worked optimally at pH 5. The optimal temperature was 50 degrees Celsius. However, thermostability revealed that the enzyme remains stable over time at 37 degrees Celsius. The kinetic parameters revealed that for starch, the Km being low shows that the enzyme is able to hydrolyze the substrate even under low concentrations. For xylem, we see that the catalytic constant but the Km value is high. So the enzyme is sufficiently able to hydrolyze the enzyme, however, not when the enzyme is not abundant. For the oligosaccharides, using LCMS, we had the major product produced being xylotriose, followed by xylobiose. With the second hydrolysis with the C118, removing the side chain substituents, we see that the enzymatic profile or the TLC profiles are different compared to that of the C118 secretome by itself. The control we see at the baseline, the substrate remaining at the baseline. With the glucuronol esterase and the furyl esterase, we have a very distinct profile, with glucuronol esterase having a very defined band close to the baseline and it having very faint bands towards the end of the plate. Similarly with furyl esterases, xylanase, being a GH10 enzyme, has many defined bands across the TLC plates. For antioxidant, for A, we look at the green bars, which present the H2O2 concentrations, the control being the H2O2 control, showing the total H2O2 radicals present. The blue line presents the antioxidant radical scavenging activity of the various oligomers with the side chain enzymes. For A, we see that with the furyl esterases as well as the glucuronol esterases, we have more scavenging abilities. With B, the blue bar shows antioxidant garlic acid equivalents, and the green line showing antioxidant scavenging. We also then here see that furyl esterases scavenge the unstable radical more. This is, according to literature, we know that the antioxidant properties of oligosaccharides are linked to those extra, those bonds formed between those double bonds with ferryl esterases and the extra bonds that can form sugar conjugates with the radicals. In conclusion, we have successfully characterized the C1184 secretome, looking at its zymogram, looking at its xylanase, cellulase, and amylase profile, enzyme profile. We also then looked at the biotechnological promise of producing oligomers with antioxidant properties, and this aligns with the United Nations goal of Sustainable Development 12. Thank you. I'd like to acknowledge the following people.
Thank you, Brittany. Well done on all your hard work. Any questions for Brittany?
Okay.
Thank you, ma'am, for such a wonderful presentation and the good research that you done in terms of characterizing starch and proteins. So within these characterizations that you were doing, is there anything you would consider not so obvious that you did?
So with this study, the secretome, the C1184 secretome, was actually purchased as a cellulase from Sigma. And so when we characterized, we saw that it had significant activity on xylane and starch substrates. So not what you would see with a cellulase, and the activity on xylane being more than that of cellulase kind of started giving us the idea that perhaps it's a secretome and not a cellulase.
So within those discoveries, did it inform you into maybe developing a better characterization framework?
Not necessarily. Something that we are currently looking into in our lab is molecular docking, which would then be more resource efficient. So yeah, that's one thing.
No, thank you. Thank you very much.
Okay.
Thank you for such a beautiful presentation. I just have some few questions with regards to your kinetic parameters, especially on you mentioned Vmax, okay? Which is your maximum
Velocity.
exactly, yeah. How was your Vmax determined? Was it determined experimentally for your enzymes?
I did it by using a range of concentrations for my substrate, and then we did it using the Michaelis-Menten formula.
Formula, okay.
Yes, and that we then used Excel Solver X to then plot the graph and then compare it based on the Michaelis-Menten formula.
So on the graph, okay, no, I get it. Thank you.
Okay.
I don't know if I missed your source of lignocellulosic material.
So we used a range of substrates because the first objective is to characterize the enzyme. So therefore looking at the different enzymes involved. So we used commercial substrates since it's an honors project. So we used a substrate from xylane, cellulase, and as well as starch.
Oh.
So they were all purchased from Megazyme and Sigma.
Oh, okay, thanks.
Okay, thanks.
Colleagues, our next presenter is Machera Letswalo.
Hello, everyone. I thought when they said this is a graveyard session, all the nerves also die off. So, as introduced, my name is Machira Parsons Litwalo. I'm the country researcher here at the CSIR. I'm from the Bioprocessing Development Group. The talk today is going to be about the large-scale production and application of biocoating product for the fruits and vegetable industry. I'm going to give you an overview of how we developed our product and also how we applied through field trials. To give a bit of a background, large-scale commercial agriculture, especially horticulture, which is the production of fruits and vegetables, is a major part of our economy. It contributes to our country's GDP and is a primary source of foreign exchange through exports. This industry currently is moving towards the use of biocoating agents as opposed to chemical waxes. This is because biocoating agents are susceptible, are sustainable, and are an innovative approach to managing our fresh produce after they've been harvested. They are edible, they are non-toxic, and they provide a safe way to protect our food. So one might ask, what are biocoating agents? These are materials used to create a film or a thin layer, a thin protective layer on the surface of fruits and vegetables. They are made from natural and food-grade ingredients, such as the lipids, polysaccharides, which are your sugars. To give a bit of a context, post-harvest degradation of fresh produce is a global issue because fruits and vegetables are susceptible to natural processes such as microbial contamination and also moisture loss. The scale of the problem is bigger than we think. According to reports, 30% of our fresh produce is lost between the farm, the market, and the consumer, which now poses a threat to our food security. The problem with the current use of chemicals by farmers raises health and environmental concerns. When we looked at these challenges, we saw that there's an urgent need for a sustainable and safe, effective approach or product, especially for the export market. So in this project, we aimed to develop a scalable bio-based preservation technology. And to achieve this, we set out two objectives: to demonstrate a scalable manufacturing process for the production of our biocoating agent, and also to validate the product's efficacy through field trials. And the expectation out of this project was to see a reduce in reliance on these chemical waxes and pesticides. Most importantly, to extend the shelf life of the produce, which will help improve the distribution internationally and nationally. So one might ask, how do we then produce this biocoating? If you put your focus on figure 1, this is a process flow sheet of how we produce our biocoating agent. So we start by inoculating a bacteria, a microorganism, in a flask. We then transfer the microorganism into a 300-liter bioreactor, which is under controlled conditions, to produce our EPS. Those are sugars. After fermentation, we harvest and centrifuge to separate the exopolysaccharides from the microbial cells. We then mix the exopolysaccharides with food-grade ingredients. It's just that based on IPs, I cannot give mention to say which ones are those. We then dry the formulate, we mill it into a fine powder, and then we have a ready-to-use product. The product met all required quality and safety specifications, which allowed us to then continue with our field farm trials. The trials were conducted in collaboration with the farmers in South Africa. Three produce, which are grapefruits, tomatoes, and oranges. The study was conducted on these three fresh produce. For the grapefruit, the study was conducted for about 90 days at a Solil farm in Hoedspruit. This is where, come back to the slide. This is where, if you put your focus on figure 3, this is where you see the schematic diagram shows you the final product, as I mentioned, is a powdered product. This is where it was mixed with water and spray coated onto the fruits, which were then dried and sorted so that we eliminate those ones that are already compromised for the purpose of our study. They were then packaged and brought to us at the CSIR for us to do our further studies. Go back. So if you focus on figure 2, it will show you that the CSR, those produce, there were 2 studies that were conducted on the food produce. One was applied with the CSIR biocoating agent. The other one was coated with citrosol, the citrosol from the farmer. From the diagram, you can see that the CSR biocoating demonstrated better moisture retention compared to the commercial benchmark. Grapefruits treated with the CSR product showed moisture loss of 21.86%, whereas the citrosol treatment batch had a 47% loss. The qualitative analysis showed the CSR coated fruit maintained better texture and had delayed decay compared to the control. So upon our analysis, we realized that in as much as it wasn't a microbial inhibition, we realized that somehow there are pathogenic organisms that are growing on our produce. However, as I said, the CSIR biocoating agent was able to delay that decay. So, trial 2 was on the tomatoes, where three batches were assessed. One was coated with the CSR biocoating agent. The second one was coated with the farmer's chemical of choice, which was sodium metabisulfate, and the third one was left untreated. From our analysis, the CSR biocoating resulted in a lower mass loss compared to the untreated and those treated with sodium metabisulfate. By day 30, the fruits coated with the CSR biocoating agent showed a mass loss of 9.33% compared to 10 and 9.8% of the untreated and the sodium metabisulfate respectively. This shows that the CSR product effectively delays wrinkling and softening. This can be viewed on figure 5, where you can see on image B, you can see the wrinkling. I don't know if it's visible. The third trial was done on oranges, where 2 batches were analyzed. One was treated with the CSR biocoating agent. The other one was treated with the citrosol, the same one that was treated with the grapefruits. So in this case, both the products didn't show any much long-term efficacy. However, from what you can see on the pictures on picture C and D, those oranges were treated with the citrosol. The pictures in A and B were treated with the CSR biocoating agent. It shows you that the CSR biocoating agent was able to in as much as it didn't assist with reducing the moisture loss, but it assisted with cells, microorganisms, pathogenic microorganisms growing on the organisms, on the fruits. In conclusion, we can say that we managed to demonstrate a scalable end-to-end manufacturing process of a product that is safe, biodegradable, and it's effective in prolonging the shelf life of various fruits by reducing water loss and microbial decay. The technology it's also sustainable. It's a sustainable alternative to conventional chemical preservatives for agricultural sector. In our future work, we are looking to conduct other large-scale trials across different seasons and on other produce like mango, pineapples, and grapes. We are also looking to perform toxicological and safety assessment to meet food safety regulations. We are also looking to explore combining the CSIR biocoating product with other preservative methods. With that said, I would like to thank you and also acknowledge my supervisors.
Well done, Machere, on a very excellent presentation. Any questions for him? Okay, we'll start with the gentleman at the back with the white shirt.
Yeah, thank you for a very good presentation. What I'm interested to know is the effect of different weather conditions on it. So if you had the coating on a tomato, for instance, when I saw in your second trial, if it was kept in a cold environment versus in a very hot environment, how would that affect the mass loss?
Currently, I think that is the second stage of when I said in the next phase of our research is to look into including other preservation methods. That's when we'll go into temperature to see if in multiple temperatures, where is it best to. We haven't got to that study yet.
All right, perfect, thank you.
Oh, I'm not the one who's called.
Okay, thank you. Actually asked one of the questions I wanted to ask, but I've got, I think, two more. I saw that you performed some benchmarking with, standard, methods. So I want to ask why only one? Is that the best in the market?
In terms of the citrus oil, because it was tested on oranges and also on grapefruits, which are from the citrus families. We saw that from the farms that we got the fresh produce from is what they were using to preserve their fruits.
Okay, so
that's why
you got it from the
yes.
from the farm.
Yes.
Okay, but did you go further to investigate what other people use so that you can at least be able to benchmark your product with the current standard with other product? That's one. But like you said, you can introduce that in the future. Yeah, thank you. The second question is the production cost, which we actually also influence the selling price.
Yes.
So how does your production cost compare to the existing ones? And
currently, I would say because we are running it at a 200-liter scale, because the major, in terms of techno-economics, you can see that the major cost comes from production, which is when we are running the 200-liter bioreactor. But because we have a system, a software, Super Pro, that is what we can use we used it to simulate, to say, if we take it to 1,000 liter, what happens? And we realized that in most manufacturing processes, when you upscale, you then reduce the cost. At small production, then I could say, the production the cost is a lot. But when we upscale, then we take down the cost.
Okay, sorry, thank you for that answer. So my last question is similar to what I had. Okay, thank you.
I'm more interested in the mechanism I'm more interested in the mechanism of action of the biocoating product. So, what component affords it the ability to maintain the quality of the produce, and what component could potentially afford it the quality of being antimicrobial?
From research, I think one of the presenters had spoken about exopolysaccharides, them being able to inhibit the growth of other microorganisms. So this is from a study to say exopolysaccharides are capable of inhibiting growth of other microorganisms. So because we had a microorganism in-house that can produce exopolysaccharides, then we thought it fit for us to try and see if we can find a solution for the agricultural sector.
So this means basically with this biocoating, which is based on the EPS, your target is actually microbes, like you're hindering microbes in order to further to allow the fruit or vegetable
Yes.
to maintain.
Yes.
Oh, okay,
Yes.
got you.
Yes.
I think just one last one. Ronald's one. Because I just see the CSIR byproduct agent. Do you care to share what's that? Or is it,
I did
or is
mention
that
in
an
the
IP kind of
Yes,
issue?
the presentation that I cannot. But there's, let's see. Yeah, it seems to say there's starch in there. Maybe you can start from there. I just gave you 25%. I gave you 25%.
All right, no.
Yes.
All right, well, thanks, man. I just wanted to.
Thanks, man, for the great presentation. I just have one question with the precursor. The precursor is, at what TRL level is this product?
Tier level 6.
6.
Yes.
Okay. So I think we are all aware of the advantages of products like fertilizers, and also some of the disadvantages that they tend to have upon consumption. So I want to understand if whether at this point in time, have you guys already looked into studies that look at the disadvantages of overconsumption of this coating product, given that you said it's all... Sorry?
Second stage.
Oh,
That will
that's
be
the
in our second stage.
Okay.
How would it affect you when you consume this much of the product? That is will be in our second stage. Yes.
Thank you again for the opportunity. Thank you for the presentation again. So my question is, following your experiment, I want to know what storage conditions did you use? Because you didn't tell us what storage conditions. And then that condition you use, is it the same with what the farmers use with the product you compared it with? Because if the conditions are not the same, it might affect your results.
Oh, yeah. No, we set out them to say, okay, what temperatures do you store your fresh produce before you export or you distribute? 19 degrees Celsius. So it was good that our labs are always at 19. So we just had to get a lab so that we can store them in there. Yeah.
Thank you, colleagues. For the next presenters, colleagues, I was kidding when I said we need to get home quickly. Take your full 15 minutes of fame. We are all here to listen to you. So with that said, our next presenter is Stefan Karamanski.
All right. Good afternoon, everybody. Today I'll be presenting on the topic using battery energy storage systems and renewable energy to reduce municipality costs in South African town case study. So just a little bit about me. My name is Stefan Karamanski. I have a background in electrical and electronic engineering, and I've also completed my master's in renewable energy engineering, and I work as a researcher here at CSIR within our energy center. And I promise you, I don't only own CSIR branded t-shirts and shirts. I've also had the pleasure of being on site a few times, and it's really been a positive experience. So these are just some of my focus areas, including wind energy, solar PV, hydrogen, smart grids, etc. Okay, so this is just a little bit about what we'll be talking about today. So I'm sure you're all well aware that South Africa is in the midst of an energy crisis. However, this has also sparked some kind of development and innovation within our country. Embedded generation is on the rise, and with renewable energy penetration increasing, we've seen an uptake in commercial and residential sectors. Municipalities have also noticed the advantages of renewable energy, and the City of Cape Town has used hydro pump storage to reduce the levels of load shedding within the city, and this has improved the quality of life and service delivery, as well as reduced economic losses and traffic congestion. So in this specific case study, we're going to look at a hypothetical scenario in the town of Hermanus, which you can see with the yellow pin on the right over there. And this Hermanus municipality is interested in a hybrid approach, including wind, solar PV, and battery energy storage systems, with a minimum 50% renewable energy penetration, and this is to meet the town's sustainable development goals. They are also interested in electric vehicle charging facilities to keep up with the latest technology, and this is quite an interesting case study because of the specific tariff structure of the Hermanus municipality. So I'm sure we've heard this story churned over and over again about load shedding, but I thought this is just quite a nice visual of load shedding over time, and you can see that we were hit pretty hard in 2023 with a lot of load shedding, and just when we thought we could get out of it in 2025, it came back to bite us. But what's most important about this is that we should be well aware that there were many times when we were on the brink of having load shedding, and it's not something that we should just dismiss for the future. So Eskom has also published data of rooftop PV. This data is a little bit old, coming from 2023, but you can see that there's a significant increase, even month on month, of the estimated installation of rooftop PV in South Africa. And as of September 2024, Eskom estimated 6.1 gigawatts of installed capacity of solar PV, which is a very, very big number. We've also seen a big reduction in renewable energy tariffs. So on screen now you can see the average tariff cost from bid window 1 to bid window 5, and we can see a reduction of 78% and 91% for wind and solar respectively from bid window 1 to bid window 5. And this picture doesn't tell the whole story. In actual fact, in bid window 5, the lowest bid was 34 cents per kilowatt hour, which is a very competitive price for Dwarselig wind facility. So not only have we seen a reduction in renewable energy costs, but we've also seen a reduction in lithium-ion battery costs. Here you see a plot over time of the reduction in cost of lithium-ion battery packs, which you can see as an exponential decrease. And with variable renewable energies being intrinsically inconsistent, batteries assist us in creating a smooth, reliable power supply. And this results in the combination of wind, solar PV, and battery energy storage, creating an appealing solution to many of our energy supply problems. So lithium-ion batteries also have the added benefit of being able to reduce costs by peak shaving, time shifting, and arbitraging, while supporting the self-consumption of renewable energy. So this is just some background on load profiles. This is a study I did in 2023, looking at the South African wind farms diurnal profile, which is just a fancy way of saying a 24-hour-long period. So we can see there a red and a blue plots. The blue one is a typical power from the 36 operational wind farms in South Africa, and on a twin axis you can see the Eskom demand. And we can see that they align quite closely with the peak time, and this also creates opportunity for solar PV to come in and fill that gap during the day hours. So just remember this demand profile, the Eskom demand profile, for later. So we have the technology, we have the natural resources in South Africa, but what about the grid connection? And this is a big problem for those of you who know about the challenges about renewable energy in South Africa. In this study, we assume that there is sufficient grid connection, despite the Western Cape, Eastern Cape, Northern Cape, and hydro regions being completely saturated at this point. So electric vehicles have also seen quite a large uptake in South Africa, and this data represents that we've seen a 67% change from 2022 to 2023, and 110% change from 2023 to 2024. And this is expected to rise for the next year. And if you've even been paying close attention while driving around on the roads, maybe you might have even seen the latest Toyota hybrid, or maybe a new Chinese EV on the road, or maybe you've even seen some of the EVs here on the CSIR campus. So this is no longer just a thought, it's actually a reality. So moving on to the methodology now, Homer grid was used for all of the modeling in the study. And the demand profile of the Hermanus municipality is not necessarily something that is easy to access or publicly available. So remember that demand profile I showed you earlier from Eskom? That is just a representation on screen now of that normalized profile. And that profile is then scaled to the population of Hermanus using census data, and then takes into account the population growth rates for the subsequent years. So we want our system to not just provide power, but we want our system to provide power reliably. And I mentioned to you about the risks of load shedding, so we also incorporate grid outages into the model, and 100 two-hour sessions are incorporated. So for each one of those black stripes you see there, that is when the grid is not accessible for any power delivery. From literature, an 87 Rand per kilowatt hour penalty is applied to incorporate any economic losses if energy cannot be served. So as I told you before, the Hermanus municipality has quite an interesting tariff structure, and as they form part of the Overstrand region, their time of use rates are used. And you can see here they have a low season rate, they have a high season rate, they have off-peak standard and peak times, and these are all incorporated into the model. The import and export rates in the model are used as the exact same figures. However, this is not really realistic because typically the export rate is a lot lower than the import rate. So in other words, you're selling back to the grid at a much lower rate than you are importing it. A 47 Rand per kVA demand charge is also incorporated, and this is to penalize users for high demand, which we will show how renewable energies can solve this. So in other words, you're going to pay for every amount of power that you receive, you're going to pay for this. So in other words, you want to minimize that. So in terms of the energy storage, lithium-ion battery energy storage is used, and the reason for this is simply because it can store large capacities, has a fast response, is able to support variable renewable energy sources, is scalable to large systems, and has improved significantly in safety and cost, as I showed you before. The Homer advanced storage battery model is used with an efficiency of 92%, with a minimum state of charge, or SOC, of 20%. So in other words, we only charge the battery to a minimum of 20%. We can't discharge it further than that, and that is in line with most manufacturers' recommendations and requirements. The degradation limit is set to 30%. So in other words, if the battery degrades to 30% of what its original capacity was, the battery is deemed no longer usable. And it's not wise to size the battery energy storage system based on peak demand, especially in this scenario, because the peak demand occurs for such a short period of time that it's quite easy to oversize the system. So instead, we look at the maximum of the average load, and that is 6.8 megawatts. So if you take that into account, the fact that two-hour load shedding sessions can occur one after the other, the SOC limit, as I just mentioned, to 20%, adding some capacity for arbitraging, and then we end up with a battery energy storage system of 39 megawatt hours. Okay, so in terms of energy generation, we mentioned before that there's this requirement of a minimum of 50% renewable energy penetration. So we have obtained solar and wind resource data over the past 22 years to look at the long-term climate of the region. The wind and solar energy are used in tandem, more specifically 6 megawatts of PV, 4.5 megawatts of wind. So in total, we end up with 10.5 megawatts of renewable energy. More specifically about the PV, in the model, the Homer generic flat plate module is used, and the Homer 1.5 generic megawatt wind turbine with 80 meter hub height. So we also included a 25 megawatt converter with zero cost and 100% efficiency. And the reason for this is simply just to not try and intervene with the renewable energy results, to try and keep those results separate to the influence of the converter cost. So I just want to re-emphasize that the reliance on the grid is not completely removed, so we can still make purchases from the grid, but we're just combining that with our renewable energy sources. Okay, so in terms of the EV charging, here you see a yearly profile. I mean, you can see where it's highlighted sort of in the middle from about 6 a.m. till 7 p.m. Basically, the day hours is where we're having the most charging as expected. And we have incorporated 7 high-quality 150 kilowatt chargers with Homer Grid's highway charger load profile with a 20 minutes average session and assumed 40 sessions per day. And we have a queuing system which is implemented, so in other words, if all of the chargers are being used at any one time, there's a queue that is created, and as soon as one of the chargers are available, it is allocated to a new user. The split for large and small EVs is 30 to 70%, 30-70, and the reason for this is we anticipate that there'll be an uptake of small EVs much more significantly than large EVs simply because of the high cost of EVs. Okay, so it's not really too critical to understand what's going on over here with the economic parameters. These are just all incorporated into the model. Maybe something of interest here is that the project lifetime is 15 years, but all of these values are taken into account in the economic modeling. That's just there for completeness sake. So moving on to the results. Hopefully nobody's fallen asleep by this point because this is the slide that hits home. So the design system has an internal rate of return of 25.3% and a return on investment of 39.5%, as well as a payback period of 5.1 years. And the system does indeed achieve the requirement of 50% renewable energy penetration, achieving a percentage of 53.9%. There's 0 kilowatt hours of unmet load, and the PV and wind capacity factors are 16 and 44.1% respectively. And the reason for this very high wind capacity factor is because Hermanus is quite close to the ocean and benefits from the low surface roughness of the ocean, so it generates quite high wind speeds in that region. The EV charging stations are also implemented successfully with 14,500 sessions per year with no unserved sessions. So most importantly, the annual consumption and demand charges savings are about 173 million and 2 million rand respectively, which totals a total utility saving of about 175 million rand. Okay, so this is just an interesting snapshot of the system. It might be a bit small, but I will guide you through it. So as you can see on the top plot over there, the red is the load, so that's the load of the municipality, how much energy they're using at any given time. The blue is the battery state of charge. So in other words, when it's saturated at the top there, it's at 100%, and you can also see at the bottom, it never goes below the 20% rule that we set out before. The yellow is the grid demand charge, and whenever there's a dip in the grid demand, that just means that there's currently load shedding happening right now. In the bottom plot, the orange is the grid purchases. The green is the solar PV output, and you can actually see the tracking throughout the day when the sun rises and peaks. You can see that the solar PV tracks that, and then the wind, which is quite erratic in nature as per expectations. So one interesting thing is that if you look over there, sort of in the middle of the screen, we can see that there's an instance of load shedding, and we can see that there is currently a demand indicated with this red. And because the blue and green insufficiently are able to supply that load, and we have no grid purchases because there's load shedding, we can actually see a dip in the battery states of charge. So the battery, in other words, is supplementing the load. And we can also see that the system has optimally decided on a limit over here where the grid purchases are saturated, and that is the optimal value that I told you before to try and save costs on the grid demand charges. So in other words, the system is working just as expected, and we can see that it is all working interchangeably and working successfully. So in conclusion, we have created a hybrid renewable energy model to serve the energy needs of Hermanus municipality with a total of 10.5 megawatts of renewables and 39 megawatt hours of battery energy storage systems, with a renewable energy penetration exceeding our requirement of 50% at 53.9%. We have incorporated 7 EV charging stations successfully, as well as a battery energy storage unit to deliver a reliable, smooth power supply with no unmet electric load. So the levelized cost of electricity for this system is 1.37 rand per kilowatt hour. And the really amazing thing about this study is that this is a flexible model that can be applied to other towns, so it's really useful for municipalities, policymakers, and town planners. So in terms of future work, I think I would like to use recorded data for more representative modeling instead of just making assumptions, incorporate the converter costs to showcase real-life costs that are associated with renewable energy systems, and use more relevant parameters, for example, more relevant financial parameters and costs of PV panels and wind turbines. Thank you very much, and you're welcome to contact me for any more questions.
Well done, Stefan, on such an excellent presentation. Can we take questions, colleagues?
Thanks, Stefan. So I imagine on your last results slide that the batteries are lithium-ion.
Yes.
How do you think solid-state battery technology would compare to that?
I'm not too sure. I haven't really evaluated that too much, and the main reason for that is simply because of the cost of lithium-ion as well as the benefits. I think lithium-ion batteries are almost the jack of all trades when it comes to batteries. I think the major concern with them was safety in the past, and we had a lot of uncontrollable fires. So I'm not sure how solid state would compare in terms of a technical standpoint, but I can imagine that it must be more expensive. And in a situation like this where we're looking at an economic driver, I think lithium-ion batteries make a lot of sense. But it's a great question and something to be investigated.
Sure. Thanks.
Thank you. Just more like a follow-up question, Stefan. So sodium-ion batteries are more cost-effective than lithium-ions, but they're not safer than lithium-ions. They explode a lot, the sodium ions. So we should find along the energy storage literature that more people are investigating sodium-ion batteries than lithium. Yeah,
Absolutely. You must also remember that cost is not necessarily tangible. So yes, you can go and buy an off-the-shelf battery here, you can go buy an off-the-shelf battery there, but there's also cost and risk. And in many of these projects, that risk is too high for a sodium battery. So that's why I think it's almost become a standard procedure to incorporate lithium-ion batteries, especially because of the EV market, where a lot of research has sort of pushed lithium-ion batteries. But I think there's definitely lots of new battery sources coming through. It's not like lithium-ion is the holy grail of batteries. You can do some research on vanadium batteries, which are also really impressive, that don't degrade over time, or very, very little degradation. They're just quite expensive, and they're currently sort of in the proof of concept stage. But I definitely think that it's battery energy storage is always something that we want to improve on, and the more energy storage we can harness, the better we can produce our grid networks and energy storage system delivery.
Sorry, on that thought, did you incorporate some of the smart grid principles in your research? Because I heard you mentioning exporting and importing energy in the grid.
Yes, absolutely. So if I actually can go back to one slide, hopefully you'll be able to see this. But you can actually see over there of the consumption. So the AC primary load is essentially just whatever the demand is. So if you're switching on your kettle or whatever, that would be your AC primary load. Your EV charges is pretty self-explanatory, and then your grid sales is 11.5%. So of the consumption, 11.5% of that is actually being sent to the grid. So this is just a showcase for municipalities to understand that it's not all about just, if you have a solar panel, whatever that is producing, you use in your municipality or your system. There's actually the opportunity to sell that back and make a profit. So it goes back to a lot of energy trading, and if you want to learn more about that, I'd highly recommend looking into VPP's virtual power plants, which is the next big thing in terms of smart grid technology. Yeah, cool. Any more questions?
Yeah, out of time.
Okay, cool. Thank you.
Our next presenter will be Given Ndlovu.
I think I might be a bit short here. Hopefully you can hear me. Afternoon, everyone. My name is Given Ndlovu from Energy Center, CSIR. I have a background in electrical engineering, and I'm a candidate researcher with primary focus on power systems. Thank you. Thank you. So the work that I will be presenting today is a pilot study that was undertaken in collaboration with ESCOM with the aim of determining or investigating the impact of embedded generators on their distribution system. So just to give a bit of a background, power systems around the world are going through a gradual shift from these centralized vertical systems where power was generated from the top and transmitted down to passive customers, as you can see on our diagram on the left side of it. And now, with the active participation of consumers where they can generate their own power as well as feeding back into the system, we have a decentralized system, as you can see on our right-hand side of the diagram. So this integration of embedded generators into the network system typically reduces power flow on the downstream by supplying the local loads first, and also ultimately reducing the system losses. However, while all this is good and well, there are issues associated with this, and some of the issues, as you can see, is the reversal of power flow, excessive voltage deviations. As we know, in terms of solar, that is not reliable that much. In the case of a cloud cover, you can easily lose power from maximum to minimum, thereby altering the voltage. And also an increase in system losses, especially if this generator is not optimally located or coordinated. And lastly, the complexity in cost recovery and fair tariff design, which I'll get to in more details later. Now, as I've mentioned, this project was done in collaboration with ESCOM with the aim of determining the impact of these small-scale generators into their network. And the reason for this particular study was that initially ESCOM assumed that all these generators are actually helping the system by reducing the power flow and ultimately reducing the system losses. However, they are seeing new trends where there are excessive losses which are caused by these embedded generators, but obviously not all of them. Looking into the losses, system losses in distribution network comprises technical losses as well as non-technical losses. So the first one, they are inherent due to the resistance on the impedance of the electrical network. In other words, they are there by nature or by the virtue of the network. And the second one is the non-technical losses, which I think most of us are familiar with. This one can occur due to metering issues or illegal connection or theft of power. But in this case, we're only going to focus on the technical losses. Now, to summarize this slide, these loss factors are accounted for in the tariff design. In other words, when you purchase energy, the losses that are occurring in the network are already included in your charge. By definition, these loss factors, also referred as loss sensitivities, represent the marginal impact of active power injection at a particular node on overall system losses. In other words, to give an example, if I have a generator here and generating a certain megawatt, how much losses can this particular generator cause on the network, or how much losses in reduction can it cause, or how much help can this particular generation, assist? Going forward, these loss factors that we've calculated can be negative and positive. A positive loss factor will mean that that particular generation is actually causing losses on the network, whereas a negative loss factor will mean that it is helping the network, as I've explained initially. Some generators, or initially all of them, were thought to be helping. However, now the trend seems to be the other way around. I will explain more. I'll get to that. Now, looking at the methodology, there are essentially two methods used to determine these loss factors. The first one being the classical load flow, and the second one being the analytical based. And not to bother with the details, in our study, we went with the first one simply because the power factory or the software that we were using was compatible with this particular one, and it was suitable for our studies. The only downside was that it can be slower, especially when the network is very huge. And the next 3 slides basically explain how we went about calculating the losses in our studies and the approaches and the philosophies basically. So what is happening here is that we have high voltage network or case files, and also the medium voltage case files. So if you look at the ESCOM network, the transmission normally goes all the way to 32 or even 22 sometimes. But in this case, we're specifically looking at not necessarily transmission, but though the high voltage network was going into the transmission, and it was starting from 22 kV all the way to 400 in most cases. And anything below 22, it was represented as a lumped load. And to give an example for this one, if you look at, for example, the city of Tshwane network and try to locate CSIR, you probably won't see it. And if you do, you're probably going to see it as a single load, which is not accurate, it's just a representation of it. And then the medium voltage case files on network, they mostly start from 22 all the way down to 400. So again, using the CSR example, we have our own network here, which starts from the 132 all the way down to 400. So to determine the impact of these generators on the overall network, we had to get a more representative network by merging the 2 case files, or two case files. So in essence, we had to replace the lat aggregated load with the detailed model. So in this case of CSIR example, you will take the city of Tshwane, remove that aggregated load, and merge it with the detailed CSIR model to get a more representative network, because that's how it is in reality. So this diagram basically highlights the methodology that we followed. Firstly, we had to merge these case files, ensure that it can converge, and then calculate the loss factors. But in this case, we had to make sure that we get the performance of the network before we even integrate the renewables. So we had to switch the renewables or put them out of service and calculate the loss factors. And thereafter, integrate or put them in service and then calculate the new loss factors. And then after that, analyze. So what's happening here, we know that as I've explained, the integration of these renewables, they actually change the set point of the network. Hence, we had to get the base without the renewables, and then after, and then compare to see whether these generators are causing losses or reducing losses. Now, this is more or less similar to what I've just explained. To determine the impact of these generators, we had to create some scenarios, the first one being the base case and the other one being the generator scenario or case. And then the diagram here highlights the actual methodology on the power factory Dix Island. So firstly, you had to select your methodology, which is the classical load flow, and then select injection nodes. So in this case, the nodes where we were injecting power to see the impact, it was the generator nodes, meaning that it's where the generators are connected. And then we had to select the equipments to monitor. In this particular case, by default, the software will select all the elements, including lines, transformers, switches, and everything. But we had to filter and only look at the branches or lines, and also select our desired threshold sensitivity. By this, in this particular case, we went with 10%. So what that actually means is that if you have a generator here and you are injecting power, we want to see the impact of it on the rest of the network, but it wouldn't make much sense to have a generator and then here in Gauteng, like a smaller generator, and then look at Mpumalanga. There might be some changes, but it's very, very minimal. So we had to cap it and say 10% is good enough. Anything below that, we neglect it, because in most cases, if you have a smaller generator here, there might be no change in Cape Town or somewhere far, but we're focusing on the nearby network. So from there, this one, we're basically choosing to evaluate the impact of these generators one by one or all of them at the same time. So in our studies, we did both, where we were evaluating each generator in 10 and also all of them at the same time. But for our studies here, we will only show for demonstration, we'll only show the first one. And then looking at the results and how to read or interpret them, remember initially I mentioned that a positive loss factor will mean that that particular generator is actually causing losses or increasing, contributing in system losses, and the negative one will be the other way around. So in this case, as I've mentioned, we had to do the study without the generators and obtained these particular values, and then do the very same study but with the generators in and obtain these. So if you look at generator number 1, or rather node number 1, because this could have been a node, doesn't matter much. If we look at generator number 1, initially we obtained a value of 0.564, and with the generator, the loss factor increased to 0.587, which indicates that the presence of this generator will actually cause or increase losses in this particular branch. And again, for more information, we chose a branch for this. So this is just one branch we are monitoring here, as I've mentioned here. And we are looking at the impact of all these generators on that particular branch. So you can see here that generator 1 as well as 4 and 5 are exhibiting the same behavior. They're actually increasing losses on the system. However, generator 2 and 6 are actually helping as far as system losses are concerned. As you can see here, we had 0.693, and now we have a lower loss factor. And then if you look at another example where at first glance you can tell that we have a lot of negative, and I've said that a negative loss factor will mean that the generators are actually helping the system. So on the base case, these are the loss factors we've obtained. On the generator case, we've obtained these loss factors. Again, if you look at generator number 1, you can see that we have a negative 0.759. However, now we have negative 0.157. So all of these generators are exhibiting the same trend, but what we are saying here is that the loss factors increased in a sense that they are going towards 0 or a positive. If we keep increasing generators here, you will end up with a positive loss factors. So in conclusion here, it means that yes, these generators are helping as far as system is concerned, system losses are concerned, but if you keep increasing generators in that particular area or this region, you will end up with a case where all of them or most of them are actually contributing to system losses. And then in conclusion, as I've said, the integration of this renewable energy, they typically reduce the power flow downstream and helping with the system losses. But these studies, the findings of the study is showing that this is not uniform all over the distribution system. And it is in line with what the ESCOM guys are seeing, these losses. So yeah, while some areas can benefit from these reduced losses, others are actually experiencing increase in these losses. And in some cases where the local generation exceeds the local demand, you will have the reverse power flow, which makes sense. If you have a solar in your house and you keep increasing, you will end up not wanting power from your main utility. You will actually be feeding back eventually. And that can, in some cases, cause losses. In some cases, it can reduce, but in some cases where the network is probably constrained already, it can cause losses. So this suggests that although embedded generation can help moderate losses, There's a tipping point beyond which its contribution becomes less effective or even counterproductive, depending on the network conditions and the balance between generation and demand. Therefore, further studies are needed to support the development of a more cost-reflective tariff structure for Eskom. So, in other words, what the study aims to do is to basically say which generators are causing losses on the system and how do we charge them accordingly, because at the moment, they are basically charged the same as if all of them are reducing losses. So for a lack of better word, sort of penalize them for causing more losses on the network by creating a more cost-reflective structure. And then for future work, this study was conducted for just one province in South Africa. So going forward, we'll have to do the study for the entire South African provinces, particularly given that there are regional differences in embedded generation as well as their penetration levels. So yeah, these findings will eventually inform policies and regulatory frameworks for managing technical losses and implementing cost-reflective tariffs. That would be my story for today.
Thank you, Given. Well done on such an excellent presentation. I will take questions. Just two, or maybe one. In that case, I guess your presentation was clear. Well done. Thank you so much. He's saying, or confusing. Anyway, our last presenter for this session is Samuel Makobe.
Good afternoon, everyone. I know that I'm the only thing that's standing between you going home and being here. Thank you so much for hanging there so tight. Thank you so much also, Chair, for the platform. My name is Samuel Makobe. I'm a PhD candidate supervised by Professor A.A. Ambushi and Dr. M.P. Sopela from the Department of Chemical Sciences at University of Johannesburg. So today I'm going to give a presentation on spatial and temporal variation in presence, level, and risk assessment of selected polycyclic aromatic hydrocarbons in water and sediment from Klip River, Johannesburg, South Africa. Throughout this presentation, I'll be following the projected overview from the introduction to the acknowledgement. So as we all know, South Africa is known to be one of the most As we all know, South Africa is one of the most water-scarce country or the driest country, ranking the 30th currently in the world, with limited water resources that are evenly distributed throughout the country. So many communities, especially in rural areas, as well as peri-urban areas, people still struggle to access safe and potable water. So I was very happy this afternoon, colleagues, when I was receiving free water. So some of our colleagues are still struggling out there, especially in rural areas. So we can see that the situation led to what we see, or what is depicted in figure 1, where residents from various regions of this country have to stand in a very long queue to access clean potable water. And then we can imagine others who have to buy water, and then also the situation for our elders who are pension dependent, who has to use that money that was meant for survival to access potable water. Besides increasing number of population that we have in this country, human activities such as the mining operation, agriculture, we're looking at increase in urbanization as well as industrialization, often lead to the pollution of the remaining water systems in this country. And this does not only affect the ecosystem, but it also affects the public health. So to address this matter, our government in South Africa have enacted a National Water Act, Act number 36 of 1998, which ensures that the remaining water systems in our country are treated or are managed with equity and sustainability. This law treats water as a national asset by making sure that there's systems in place to protect the environment as well as human needs. And this is made sure also by making sure that there's enough clean water set aside for basic use as well as ecological protection. Klip River, which winds through Soweto and Lenasia, located in the Gauteng province, as depicted in figure 2, which feeds in the main Vaal. This river, it serves as a vital water source for local communities. We're looking at the livestock around the area as well as the whole ecosystem. But the presence of anthropogenic activities in the vicinity of this river, such as industrial discharges, we're looking at leaking sewage systems around Soweto, we're looking at irresponsible littering. This might lead to the pollution of this water system. Hence, it is imperative for us to conduct an environmental study to determine the identity and the level of concentration level of emerging environmental pollutants in this river. And then the polycyclic aromatic hydrocarbon, abbreviated as the PHs, these are organic compound composed of 2 or more fused aromatic rings. The origin of these compounds include anthropogenic activities such as industrial discharges, burning of fossil fuel, we're looking at irresponsible littering, sewage discharges, as well as oil spills. These anthropogenic activities, they are pollutants of concern because when they're in contact with animals and human beings, they result in carcinogenic effect, and then some of them, they result in reproductive failure as well as developmental malformation. So 16 of these priority pollutants, as depicted in figures 3, have been categorized as the priority pollutants by the United States Environmental Protection Agency. When in contact with human beings, they result in carcinogenicity, neurotoxicity, as well as genotoxicity. And then when these compounds are in contact with aquatic animals, we see developmental malformation, acute toxicity, and over time, this leads to a species extinction. And then when they are in contact with the crops, we'll see the reduced crop productivity, salinization of the soil, as well as biodiversity loss. Then the aim of this research was to determine the identity level, sources, as well as the toxicity of these contaminants in water and sediment. The specific objectives include evaluation of concentration level of these pollutants into different seasons. We also wanted to optimize and extract these pollutants in water and sediment, using liquid extraction method, and then in sediment we used microwave assisted extraction. We also want to know if our method were validate, so we confirmed the accuracy of the method applied in extraction of these compounds in water using the spike method, and then in sediment sample we use certified reference material 104 for sediment sample. We also want to further analyze our sample using gas chromatography flame ionization detector, and then we apply a new study, environmental toxicity study, because we don't want to tell people that the water is contaminated, then we leave it like that. People want to know how does that affect them. So what we did, we introduced the new study to evaluate the ecotoxicological effect of the whole water and sediment using the zebrafish embryo development test. Figure 4 shows the exact location of the sampling site where our samples were collected in Soweto and Lenasia in Johannesburg. Of course, we have to do the hard work, go there, collect the water and sediment. I bet this is the very bad job. No one will like it, but over time we get to be in love with it, colleagues. Then for sample preparation, prior to collection of our samples, water and sediment sample, we evaluated the measurement, we evaluated the water quality parameters to understand the quality of water. Then after that, we collected water in those brown amber glass bottles, 2.5 liter, and then the sediment samples were collected in transparent glass jar. Then we took our samples in a cooler bag to the laboratory. When we get to the laboratory, we have to refrigerate our water sample at 4 degrees Celsius, and then we freeze-dried our sediment sample using a bio-based freeze dryer. Then we grounded them into a fine powder using an agate pestle and mortar. Then we sieved them and analyzed them in a microwave-assisted extraction using 30 milliliter acetone and hexane in 1:1 ratio. Water samples were then subjected to Buchner funnel filtration. Then we performed a simple liquid extraction by mechanical shaking using 3 times 60 milliliter of dichloroethane. Then for method validation, we employed certified reference material 104 in sediment sample, and then we used the spiking method to confirm the accuracy of the extraction method in water. Then extract of water and sediment sample were then collected and analyzed using an Agilent Model 7820A gas chromatography flame ionization detector under the following depicted optimized operating condition in table 1. Then we performed the toxicity test. This work is very difficult. At some point, you are an environmental analytical chemist. At some point, now you become a fisherman. So yes, we have a facility in Northwest. We call it a Bioaquatic National Facility in Northwest University, where we have our zebrafish embryo, and then the zebrafish who are confined in different housing tanks. So we choose this fish, zebrafish, because of it shares over 70% morphology with human beings. This implies that upon exposure to contaminants, the way they respond has a probability over 70% similarity with human beings. So this zebrafish embryo were collected, and then we separated them into male and female into 2:1 ratio. So the same as human population, more women than men, right? And then after that, colleagues, we are able to separate the two. So the one that has a belly, which means it's carrying eggs, right? That's a female, and then we have a male at the top. Then we took them, we inserted them into the ice pond tank for mating purpose, male at the top, female at the bottom. Then we switch off your light. You come tomorrow, upon separation of this net, we separate them in this net, then we take out that net, and then they start mating. After mating, we collected the zebrafish embryo at the bottom of an ice pond using the Petri dish. Then using this microscope, we are able to separate fertilized embryo from an unfertilized embryo. Then we took fertilized embryo of the same cell stage, then we evaluate endpoint such as mortality, hatching rate, and malformation. We were able to detect the presence of 16 pHs, including 4 internal standards, using gas chromatography flame ionization detector based on different retention times, as depicted by the chromatogram in figure 8. Then we used the internal standard calibration method to construct calibration curves for the concentration determination of each pH. So in figure 9, we're showing an example of calibration curve obtained for fluorine. And then the following analytical figures of merit were determined: coefficient of determination, which shows good linearity. We also evaluated limit of detection as well as limit of quantification. We looked at percentage recovery of our method, which shows good and acceptable recovery. We were also concerned about the precision of our method, and we evaluated standard deviation as well as percentage relative standard deviation. Figure 10 shows an example of analysis, example of chromatogram obtained from an analysis of extract of sediment sample. In the first picture showing the presence of pHs, and then the other figure is the chromatogram obtained from analysis of an extract of water sample showing the presence of pHs. The presence of peaks other than that of pHs here, it indicates the sample complexity of our sediment compared to water samples. Then we were able to determine the concentration of these pHs, polycyclic aromatic hydrocarbon, at each site, also the combined concentration of all of them during the low flow season and during the high flow season. So we were able to also note that the concentration level of these contaminants were higher during the low flow season compared to the high flow season. And then we were also able to detect sediment site with elevated level of pHs, such as site 7, a reference site in the low flow season, and then we've seen the observed, and the lowest was recorded in site 4. And then when we go to the high flow season, we have observed temporal variation in concentration level of this, which might be also attributed to different activities that were happening at that particular site. So we observed that the concentration changes. Elevated concentration was in site 6, and then the lowest was observed in site 3. And similarly, we also evaluated their concentration level in water during the low flow season and high flow season. So we are also able to detect site with elevated level of pHs in the low flow season and the high flow season. And then we also noted that the total concentration collected in all sites during the low flow season were higher compared to the high flow season. But it's imperative to note that the highest concentration of these pollutants were found in sediment sample compared to water samples. Then we further conducted the toxicity study by exposing our embryos to sieve sediment sample that we collected in the river. Also, we exposed them to water sample, and the test was conducted under two controls: the negative control indicating the medium, clean medium, and the positive control indicating medium with toxins. So we first monitored the hatching rate, because our zebrafish embryo were expected to hatch naturally at 48 hours post-fertilization, indicated by the red dotted line. If you look at the embryos exposed to the negative control, we've seen normal hatching rate above 50%, but when you expose them to the medium with toxin, hatching rate was affected as it was below 50%. If you remember the site with elevated level of pHs in sediment sample, site 7 during the low flow season and site 8 during the high flow season. So you can see that embryos at that particular site, at those particular sites, did not hatch at 48 hours post-fertilization. Sorry. Did not hatch at 48 hours post-fertilization, and hatching started at 72 hours post-fertilization. This means that the presence of pHs at elevated level negatively affect or delay the hatch rate of a zebrafish embryo. And the same trend was observed in water sample, but in water sample were not negatively affected compared to the sediment sample, showing the presence of less pollution, pH pollution in water samples. We also evaluated the mortality of zebrafish embryo under two control. So we can see that in a clean medium, we have less mortality of less than 10%. And then when we expose them to the medium with toxin positive control, we have around 30% of our embryos were dying. If you look at site with elevated level of pH, site 7, during the low flow, and site 8, we can see that the mortality was increasing with exposure time, meaning the more you expose the embryos to those sites with elevated level of pHs, they were dying with time. And then at the end of our test period, it's also imperative to note that all of embryos exposed to site 6 and site 7 had died. So the same trend was also observed in embryos exposed to the water sample, but the level of pollution in water sample was so less, so you can see the mortality was less, which means some of our embryos were surviving in water samples. And then we went further and evaluated the total malformation overall. So malformation over 10% was regarded as significant, which means it's site of concern with regards to developmental characteristics of this embryo. So we can see in the sediment sample, only site 9 during the low flow season was insignificant. So all other embryos exposed to these sites of sediment sample were experiencing different developmental malformation. And if you look at our sites of interest, site 7 and site 8, you can see over 80% malformations were observed in those embryos. Similar trend was observed in water sample, but because water samples were less polluted compared to sediment sample, some of our embryos didn't exhibit significant malformation from 24 hours to 48 hours post-fertilization. At 72 hours post-fertilization to the 96, that's when they started exhibiting significant malformation. Then we went and evaluated the heartbeat per minute of this zebrafish. We want to know if ever the heartbeat was not affected. So we exposed them to different media, including negative control and positive control. So we can see in a clean medium, our embryos are surviving, but in medium with toxins, our embryos are getting affected, especially in sediment samples. So I've also included a video, guys, that we made using a dinoscope showing you that how we did integrate that part and determine the heartbeat of the zebrafish embryo. We went again and determined the blood flow activity, because if the blood is not moving, they'll get to die. So we can see in sediment sample, the blood flow was affected, and I've also included a video there showing how we selected a portion in zebrafish and determined the blood flow circulation of those zebrafish. And lastly, we evaluated the burst activity. We want to know the movement, because if this site is polluted, you need to move to the other side. Also, this is the survival skill. So we can see that in the negative control, our embryos were able to move effectively, but look at this, in certain sites, our burst activity, the movement of our zebrafish embryo were really affected. So I've also included a video here. Every time it gets to a certain peak, we're able to calculate the burst activity, the movement of this zebrafish embryo. So I hope, colleagues, you can see them moving there. We use the stereoscope control, remote control, and we're able to detect those things. All right, in conclusion, we're able to observe that anthropogenic activities present in the close proximity of the river leads to high pollution. leads to high pollution in water and sediment sample, leading to the presence of pHs at elevated level. We are also able to determine that the low flow sediment and water sample had high concentration level of pHs compared to high flow. And then we are also able to see site with elevated level of pHs during the low flow and high flow season in water sample and sediment sample. And then from the toxicity test, we're able to also see that highly polluted site with pHs leads to low Hessian rate, high malformation, and then high mortality. So in future, we want to look at other emerging contaminants, such as the inorganic, especially the inorganic part, such as potentially toxic element. We can look at pharmaceutical microplastics, as well as any other endocrine disrupting compound. So I'd like to acknowledge my supervisors, Prof. Wepner, also from the Department of Zoology in Northwest, there's the guy responsible for the zebrafish facility, and then the analytical chemistry group at UJ, the funders, CSIR, NRF, Water Research Commission. And lastly, it's imperative to know that this work has made a significant impact, scientific and public contribution. So we are able to publish part of this work, sediment analysis, in February. And then this work also has received a wide range of media coverage. So if you want to follow the discussions that were done in different platforms, you can click on those links. I thank you all.
Well done, Samuel. Any questions for Samuel?
Thank you. So I wanted to know, the test was done with zebrafish.
Sorry?
The test was done with zebrafish on the variety of issues in the Klip River. So what other fish are native to the Klip River, and how are they managing in that environment at the moment?
We're not particularly looking at different kinds of zebrafish, but we know that the genome of zebrafish is highly conserved in animals, also in human beings. So particularly when I was saying that prior to extraction, prior to sampling, we look at water quality parameters. So such as total dissolved oxygen, total dissolved all solid, you also look at the pH of the water and all that. So the total dissolved oxygen was very less, showing that the aquatic animal were really affected, which means it's hard for them to breathe. And we also noted a high concentration level of the solids. In addition to that, a lot of people who are fishing in selected sites that we considered were also complaining about the amount of fish that they were getting, because those people have been staying there for a very long time. So they used to go there at this particular time and get a lot of fish. But nowadays they're not getting anything. So which means that the survival rate of those aquatic animals at those selected sites are highly affected. I hope I answered your question.
Yeah, thank you.
Thank you, sir.
Thank you, sir. Any challenges did you face while conducting your research?
I faced the challenge. Okay, the first challenge was with instrument, because I was concerned about the sensitivity. So, and then we had our instrument breakdown GCMS, because I wanted GCMS or TOF-MS, looking at the sensitivity. Then because of the research has to move, then I used the GCFID. Secondly, because of I'm wearing glasses, then during the what do you call this, during the mating and all that, remember having to walk in the lab and then it's dark and then it's steamy. So you get all of that, it's like driving a car when it's raining. So my glasses were very, 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 very harsh. So I couldn't see anything. But at some point, obviously you have to stay strong and then conduct the toxicity test. Thank you so much.
Thank you, colleagues. Can we just give one last round of applause to all our presenters? Thank you, colleagues. We are now heading towards the end of the session. I just have a few announcements to make. For tomorrow morning, I've been asked to ask all the colleagues to make sure that they are all here tomorrow by 8:30 so that we are all seated for the CEO's keynote address tomorrow. And then with that said, I would like to again thank all the presenters for the excellent work that they did today. Thank you to you all, ladies and gentlemen. You have been such an amazing audience and you made this session very engaging and very interesting. And for that, I thank you. Last one, as we leave here, can we all head towards the posters and the exhibitions? And then with that said, I call this session to its close. Thank you.